Genetic engineering is a fascinating concept which has been widely misused by media and literature. But to put it simply, genetic engineering allows us to modify the life forms and create living systems that has never before existed. This is how Light Bio engineered the living petunia that glows in the dark, or how a team of mad researchers in Boston brought back the direwolf. Just the magnitude of it can make my head whirl. But what does it take to actually become a genetic engineer and do all this fascinating work that our colleagues are doing on a daily basis? Well, you're in the right place, because here in this video you'll get one step closer to becoming a real genetic engineer. So stay tuned and watch till the end.
On tonight's program, ladies and gentlemen, we have something that's going to make you sick.
Everything begins from the core of life science, the central dogma of biology, which is DNA to RNA to protein. These are the core structures of every living life form. Let's begin with DNA, deoxyribonucleic acid. DNA, as we know it now, is comprised of 4 nucleotides, A, T, G, and C, adenine, thymine, guanine, and cytosine. A is always complementary to T, and G always complementary to C. The variations of these nucleotides furthermore guarantee the diversity of genetic materials. DNA is double helix, and the strands are antiparallel to one another. The correct reading direction is always 5' to 3', in this case left to right. On the other hand, RNA, ribonucleic acid, is single-stranded and is made out of 4 nucleotides, adenine, uracil, guanine, and cytosine, so RNA doesn't have thymine and instead has uracil. But we don't have time to explain all this. To put it simply, you have to understand how transcription and translation works, how DNA is transcribed to RNA, and how RNA is translated to protein. This is a central dogma of molecular biology and life sciences. And now, let's talk about plasmids. What is plasmid and why is it so important for genetic engineering applications, you might ask? Well, for the most cases, plasmid is exactly the DNA construct we modify to express new proteins and enzymes. It's a circular piece of DNA naturally found in prokaryotic cells and sometimes in eukaryotic cells as well. Plasmids exist separately from cell's genome in cytoplasm, so it's a distinct entity from genome. But it's still not enough. This is just the tip of a monstrous iceberg. In order to fully understand and engineer the plasmid sequences, you need a tool to visualize and modify them correctly. For these applications, there are numerous genome editing softwares such as Benchling, Genome Compiler, and SnapGene. These softwares allow you to engineer plasmids in a way that's truly transformative. Here, you'll be able to visualize the distinct components of plasmid DNA such as multiple cloning sites, selectable markers, and genome interest, and many more. I simply can't explain all the details in this video, so I'll leave some really helpful textbooks down in the description. And here comes the practical part of bench research. In programming, almost every beginner starts by the same hello world command, and biology is not any different. The first genetic engineering experiments always making a bacteria glow green under UV light. This is the basis of synthetic biology. You simply order plasmid from genome synthesis company and put it into an E. coli. If the transformation is successful, your E. coli will glow green under UV light. As simple as it is, the fact that we can simply engineer never before existed life forms is a bliss. And here comes the hard part. Well, at least for me. The cloning. Yeah, it does sound captivating, but you're not cloning sheep or dragons. No, not yet. The cloning we're referring to is a manual plasmid assembly process which allows us to cut plasmid DNA and introduce new sequences, all in ideally 1 or 2 reactions. There are multiple types of cloning methods. A traditional cloning, which is in my opinion the least straightforward and most time-consuming. Golden Gate cloning, which is the most effective cloning method so far, and Gibson assembly, which is the most expensive. All the details about cloning methods will be in description. Alright, we now know how to transform an E. coli and express new proteins, but I believe that the most valuable learning experience is by doing. And to do this, you need chemicals, reagents, pipettes, thermocyclers, gel electrophoresis machine, and many more. But as an amateur biologist, there are not many places which will actually sell you this. So, where do you actually get this? The Odin has a wide variety of kits and reagents, and it's specifically for amateurs, biohackers, and beginner professionals. Besides Odin, I recently discovered that there are shit tons of resources for cheap on Alibaba, Pinduoduo, and AliExpress. and they don't actually care whether you're in academia or not. So do your due diligence and find the best deals. Well, if you come so far, congratulations. You are now one step closer to becoming a real genetic engineer. This is the culmination of part 1. In part 2, we'll cover gel electrophoresis PCR, PAGE, sequencing technologies, and genome synthesis. So don't forget to like this video and subscribe for more.